I'm today going to talk to you about the spatial phenotypic solutions and how it can revolutionize your research. When we talk about multiplex immunofluorescence, you may be thinking of an image like this, where you can see morphological differences. The red here may be a tumor region, where the rest of it may be stroma. But the important thing is what lies beneath, or what are you missing in your data? How can these cell types and their interactions can be quantified and better understood while probing such a complex microenvironment? And in recent years, there have been more and more publications that are coming out. As you can see in this slide, there is this cartoon showing 13 different cell types, and on the right, there are the publications which say that there is a need for multi-marker spatial analysis when you are trying to understand a complex tissue microenvironment. Just by using previous conventional technologies may not give you the spatial answer that you are looking for. And these papers that have been coming out more and more in recent years suggest that. Well, we know there is the gold standard of conventional IHC and quantitative genomics or proteomics, but they lose the tissue architecture when you do this kind of studies using genomics and proteomics. So that's why there is a spatial revolution, which started with the bulk analysis of DNA and RNA, moved into regions of interest based analysis, but then at AKOYA Biosciences, we give you a true single cell spatial biology, because when you can image the whole tissue on a slide, a whole slide imaging, you can map every cell and analyze every cell present in it. That's the spatial analysis revolution. And when you ask the question, what is spatial analysis? What can we do with spatial biology? You look at this arc where you start with phenotyping, then you go into the rare cell discovery path. Are there any rare cells which may be thought as noise because you are not visually looking at the whole tissue? Are there spatial neighbors? Can a cell behave differently at different neighborhoods? Is there a multi-omic aspect to the chemokines and cytokines that are expressed? Can those be attractant for immune cells, or those be resistant or deterrent for immune cells? And finally, once you get spatial signature, you want to look at clinical outcomes. And to do that, you will need speed and high throughput. And in AKOYA Biosciences imaging and image analysis platform, we offer the whole solution. We start with our PhenoCycler and PhenoImager-Fusion combination that does discovery and translation. And then we move up to PhenoImager HT, which does a high throughput scanning of multiplex slides. I just want to let you guys know, if you were familiar by the old branding name of our products, the PhenoCycler used to be called as Codex, whereas PhenoImager HT was called Vectra Polaris. So in University of Michigan website, you may see Vectra Polaris. That's PhenoImager HT. We did a rebranding last year. So today we'll start with the combination of PhenoCycler and PhenoImager-Fusion, how a microfluidic box in combination with a multispectral imager can give you ultra high-plex data. And when we look into our arc of spatial biology, we are going to see how phenotyping can lead into rare cell discovery. This is an example of a DAPI image of a stage 2 breast cancer adenocarcinoma. And now, after the DAPI image, we have put in some markers on it. You can see lamin B1, E2F1, E category. Now, if you want, you can zoom in to more closely visualize the biology, or you can keep putting different markers. And you see that same piece of tissue you are imaging, but with 3 different markers or 4 different markers added, it is showing you a new biology. And the great thing is that this is quantitative. You can make a t-SNE plot, then you can come back to the tissue and exactly locate, oh, these are the Ki67 cells that I'm seeing in my t-SNE plot. That's where they are located. What are the other things around them? And this image led to a great discovery of rare cell types. So there were 36 biomarkers in this panel. There are 63,000 cells in this image, out of which 446 basal cells were found, and from which 44 keratin 8 and 14 positive cells were detected. When you are doing a big genomic study, that can be just a noise. But here you can see exactly where the cells are located. And what happened, the researcher Kai Kassenberg's lab, they went back and looked into their previous genomic studies, and they found those cells they saw, but they ignored. But now, with more research, they have found that these cells may have a stem-like property. So this can be a new discovery to look at healthy breast tissue and see whether these cells are present, whether the number of the cells, the position of the cells, the location of the cells can have a further outcome. And the most important thing is you have to visualize and find those cells before you know they exist. So if you are picking up regions of interest, there's a greater chance you miss this kind of discovery. Now, cellular neighborhood is an important and very interesting concept. It's almost like us. We are a different person. We are in the university, going to the lab. We are a different person when we are with our family. We are a different person when we are going out with our friends. And the same thing happens to the cells. If you look at in this image, you can see we are looking at few neighborhoods right here. The main important concept is the same cell, but a different context can change the total understanding. If you look at the cluster 1, the red CD4 T cells are most likely to be proximal to other CD4 T cells. If you look at the cluster 2, right here, this neighborhood 2, the CD4 T cells are most likely to be adjacent to blue CD8 cytotoxic T cell. But if you look at the neighborhood 6 or 7, CD4 T cells are most likely to be adjacent to yellow luminal cells, or in the neighborhood 7, it's near the epithelial cells. So can this neighborhood determine the function of the CD4 cell? And that's the important question you have to ask and you have to understand. And to do that, you need an ultra-high-plex technology, a multi-marker approach, and a single-cell resolution. That is very important. This brings us to the technology at heart that creates these beautiful images, lets you analyze. Our technology is an end-to-end solution. You start with barcoded antibodies, then you move to the PhenoCycler combined with Fusion workflow, and finally you get a beautiful image. And the beautiful part of this beautiful image is you can analyze it using any image analysis workflow that you are used to. The file is a Cupitive extension that can be used to do image analysis using any third party, any free open source software that is available. So we haven't blocked the analysis in that way. We have kept it open. So these are some of the consumables that you need to start. You get a staining kit, you get the barcoded antibody, you get the reporters where you have a fluorophore attached to a antisense barcode, and you get barcodes. That means you are not limited to a panel of antibodies that we are providing. Any validated antibody that works in your lab, works in simple IHC experiments, can be put on a barcode and put in a panel to work in PhenoCycler. The basic chemistry is that each antibody is conjugated to a unique oligobarcode. The 3 spectrally separated dyes are used here that allows clear signal detection. And during each imaging cycle, 3 reporters goes and finds its complementary barcode on the primary antibody, gives out a fluorescent signal, and that is imaged. This is the very basic of the technology. This technology, some of you may know, came out of Dr. Gary Nolan's lab in Stanford, and at AKOYA, we are the people who are commercially getting this available to all the researchers like you. The step 1 of the workflow is the manual step, where you take all your primary antibodies and you incubate into the tissue. Although this step looks like a single step, but there are in between intermediary steps. If you're using FFPE, you have to do the deparaffinization, you have to do antigen retrieval, then you do cross-linking. And this technology is both compatible to fresh frozen and FFPE samples. So once you got all your primary antibodies, for example, here in this cartoon, 40 of them onto the tissue, we have a flow cell that you put on the top of the slide. I just want to mention that this slide is just a simple 1:3 histology slide, positively charged, so that you can have the tissue on it. You put the flow cell through these holes in the flow cell, the fluidics will be going through. Then you put the flow cell in the flow cell slide carrier. The slide carrier goes into PhenoImager-Fusion. There are tubings which is connecting the PhenoCycler to the Fusion. Now, once the slide goes in, the next step is automated. You select run and the instrument runs. What happens that the PhenoCycler or the codex box sends in 3 reporters and a DAPI counters chain. The reporters find their antisense oligo and hybridize, while DAPI lights up the nucleus. You can see the fluorophores that we use in each cycle are kind of same. We just alternatively use AF488, AF750, depending on whether the marker is a low expressor and how much autofluorescence can affect on it. So if you have a low expressor, you go to the AF750 in the near infrared range. We do the imaging, which is we are calling cycle 1. Then the PhenoCycler is going to send in an isothermal buffer that will dehybridize and remove the reporters and DAPI. So your slide goes back to the same thing, and you keep on doing this reveal, image, remove, and repeat. And that's how you create the ultra high-plex image of hundreds of biomarkers. This is the software architecture where you have a experiment designer that can be downloaded into any desktop that you have. And you can create your experiment, go up to the machine, load it up into this imaging software. The imaging software has a place to put in your email. So you get notifications, all the cycles are moving perfectly. If there is an issue, you will get an email to know. And finally, we have created an open source analysis pathway for all our users who are adopting this technology with QPath and Cytomap. So with this technology, you can do ultra high-plex, single cell subcellular resolution, whole slide imaging, high throughput, and you can do multi-omic. And we are going to talk about that in a little bit in the next few slides. This is just an example of looking at how the panels look, how you can divide the panels. This is an image of a human tonsil, but you can see all other panels that have been used to elucidate different tissue. This is an example of a mouse fresh frozen panel right here. We have a lot of neuro-human FFP panels too. And not only you can do whole slide imaging, you can do TMAs. This slide shows like, I think 48 or 50 TMAs. You get single cell resolution. That means you can look at the whole tissue, but you can also zoom into the areas. You can change layers. Like if you want to look at the single cell resolution of like CD66, CD44 here. If you want to look at the T cells only, if you want to look at the B cells, and this interactive data set is uploaded in our website where you can go and click on the Minerva display and look into these data sets, how the analysis was done, the image quality. And you can get this kind of spatial connectivity, how each cell is related to each other while you do the analysis. This is the few data sets that I'm saying that's up on our interactive website. So when you are analyzing this tissue, the important conundrum that people had for a long time is how many ROIs makes it unbiased? Maybe 5, maybe 15. But there are always populations of cells that you are leaving out before knowing whether they're important or not. So with PhenoCycler-Fusion, you can do this kind of whole slide analysis. And if you look at this image itself, it has a diversity of different tumors. And when you do a whole slide imaging, you can see the heterogeneity that comes up with more of the cell infiltration versus metabolic marker expression in the same piece of tissue. And that's what we want to give all our researchers, that they decide the question. They don't leave anything out. They get everything in. And the important thing with imaging is analysis. So you can use our analysis platform. You can use the open source platform. You can use your own home cooked platform. You can use a cloud-based platform like Enable Medicine or a subscription-based platform like Visiopharm. You have the flexibility to use what you want so that you can do your data processing, cell segmentation, quality control, then do a clustering, create a UMAP or a t-SNE. If you're from the world of genomics, you know t-SNEs and UMAPs better. And then you can do the spatial analysis. That means all the cell clusters that you look at in your UMAP or t-SNE, you can go back into the tissue, find exact location and see whether there is a significance of their location, all the other cells around it on the tissue. People generally do hypothesis-driven versus discovery kind of approach, where when they have hypothesis-driven, they know the expression of their markers. They can do thresholding and gating and use like a machine learning kind of approach. Whereas when they do discovery, they do unbiased clustering, and from there, they can get into the data. So as I said, with QPath, we have this Stardist segmentation, cell phenotyping, and cytomap to do neighbor analysis, the pipeline ready. Thresholding and gating is a good approach, but there can be disadvantages in accuracy of segmentation and other things if you correctly don't know the expression pattern. With a hypothesis-driven approach, you can tell the machine learning software or the AI software that, okay, this is the tissue, let's classify it, then use DAPI or the nuclear stain to do the segmentation with cytoplasmic markers, and then you can do the phenotyping and look at the nearest neighbors, touching cells. While you're doing unbiased spatial approach, where you do clustering and phenotyping, you can get this cluster, look at the heat maps, how each population is interacting. You can do the cellular neighborhood analysis. You take one cell, anchor it, and look around it to see what different cells are. Then you take another cell, and this is how you create your neighborhood clusters. For example, if you look at the CN1, you can see those are more crypt cells, where the cellular neighborhood 5 is more epithelial cell, and you can look at the image and the quantification back and forth to understand. PhenoCycler has been widely deployed. These are some of the few examples of the tissues where we have used the PhenoCycler technology to reveal. Now it brings us to the multi-omic or the spatial functional state approach. Can you use this instrument to look at RNAs and protein? And I'm going to tell you two ways of RNA detection. The first one is the RNA-scope assay. If you have done RNA-scope assay, you know it's a manual process. It takes a week to do, where you do 3 probes at a time, image it, bleach it, go back. Now you can use the PhenoCycler-Fusion platform to do it automated with ACD Biosystems. Very soon we are launching a HiPlex 12 kit where you can do 12 RNAs on the PhenoCycler system automated. These are a few of the things that have come up with this, our collaboration with ACD, that you can start with the 12 user selected RNA-scope probes. That gives you the flexibility. You can design your probe panels from the 40K probes that are available in ACD's expert probe design software. And this is how the thing will look. So you put in your slide and it will do the cycling. I'll show some of the images. This is a HiPlex RNA-scope image with the ACD HiPlex 12 V2 assay slides. It's a tonsil sample. I can show you a 2.6x zoom in and also a 10x zoom in. You can see that the T4 probe for HPRT1, which is a low expressor, can be still picked up with this kind of imaging. It's a 40x resolution image. This was the whole slide, and we just zoomed into an area of the whole slide. Now, this goes like the RNA scope aspect. If you want to see more RNA, you don't want to get limited by 12. Then we have the multi-omic approach, where on the same piece of tissue, you can look at hundreds of protein and hundreds of RNA. I like this pizza slice tissue. I hope people are not getting hungry looking at it. So if you look at this image, this is an image where you have both protein and RNA. And we'll select a few regions to see whether, when you look at the protein signal or the RNA signal, whether we can see something new in the biology. So if you look here, we are showing the tumor region, and the RNA probes are highlighting the 3 core layers of the epithelium: the basal layer, the prickle layer, and also the granular layer. And If we now put in keratin 10 overlay of the protein signal on it, you can see it kind of creates a boundary between the tumor and the connective tissue surrounding the whole epithelium structure. In the region 2, it happens the same way. But when we look at the region 3, we can see there are portions here that are chewed up. And if we put the keratin 5, you can see something is happening in this region. To understand it better with the RNA and protein, if we add a protein like a CD68, you can see there is some new biology is waiting there to be discovered. So when you're looking at this kind of multi-omic approach, it would be important to understand these images better with looking at the chemokines, cytokines, and the proteins together on the same tissue. In the spatial journey, we are talking till now about the discovery side with ultra high plex. Once you have discovered your markers, your spatial signature, and you want to do high throughput studies, we come to the next solution that I showed in the slide way back in this presentation. This is a study where they looked at a meta-analysis of 8,000 patients, 10 tumor types, and they saw if there is just 3 markers in a multiplex immunofluorescence, it is a better predictor than all the other technologies that are listed here. This was a study done in Johns Hopkins where they data mined the publication. This is a publication that came out in 2017 from Dr. Bernie Fox's lab, where they were looking at an FDA-approved drug, Pembrose, action. It was found that out of the responders, only 41% responded to the drug when it was introduced to the patient cohort. And out of the non-responders, there was still 13% of the people who were still being able to get into the immunotherapy. So what was the problem? How single marker analysis was not able to predict the correct cohort? What they did is, first they looked at multiple markers and they divided the tissue into tumor and stroma. The results were very interesting because they saw more FOXP3 positive cells were present in the patients where there was resistance. They were like, this doesn't match up. So they did like a nearest neighbor study. They drew a 30 angstrom diameter and they wanted to look whether the CD8 cytotoxic T cells and Tregs are together has a different outcome or they are away from each other has a different outcome. And that's how they create like an immunosuppressive score where they saw that when the CD8 T cells are near the Tregs, their actions are suppressed. So those patients are not going to be on giving good results on immunotherapy, whereas in the patients where CD8 T cells were near the tumor, that was where patients were doing better with immunotherapy. And these kind of studies have been done together with the PhenoCycler and the PhenoImager HT, where you can see they deployed a 56-plex panel to look at the spatial neighborhoods. Then they created spatial signature and created a spatial score so that even before the patients are sent to a treatment, they can be stratified correctly to responder and non-responders. And the PhenoImager technology, this is a paper from University of Michigan, from Dr. Frankel's lab, where they looked at mouse tissue using a 6-plex 7-color assay, looking in TMAs, try to investigate colorectal cancer for the liver metastasis of 177 patients. People have looked into exhausted CD8 T cell anti-PD-1 therapy. People have looked at mouse FFP brain tissue using the PhenoImager technology. And people have done multi-omics in a way they have done single-cell RNA-seq using, and then they looked at our PhenoImager HT and tried to see whether those cells are associating. People have used site of mass cytometry in pediatric glioma and the PhenoImager HT in parallel to see the 3 different types of T cells where they are located in the tissue. The PhenoImager HT, or which is called Vectra Polaris before, has also as an end-to-end solution. You start with the staining with opals, then you do the imaging with the HT, and then you do the analysis using the informed machine learning software. Here, the staining is done offline, manually, or you're using a auto stainer where you do an iterative staining, and this is based on the tyramide signal amplification technology, where you send in a primary antibody, and as you can see in this cartoon, you can use the same primary antibody for probing each protein. Then you follow up with a secondary antibody which has a HRP tag to it. You add the tyramide signal amplification and opal reagents, which will deposit the opal on the protein. And then you use heat to remove the primary and secondary conjugate. So you keep on repeating this for all your antigens, and you create your multiplex slide. So the opal reagents can be available in 479 kits. There are different pan-cancer kits. And recently we have launched some PhenoCode signature panel where we have created panels to answer the question whether the tumor is hot or cold, whether there are proliferating cells and lymphocytes. But you can add one of your own marker to this panel and do a quick optimization of those panels. And the important thing is that in the previous technology, we are just imaging 3 biomarkers. Here we are imaging 6 or 8 biomarkers on the tissue. That means there is a lot of crosstalk between the fluorophores. And if you are using a immunofluorescent microscope, which uses just based on the excitation or the emission peak of the fluorophore, you won't be able to decipher a lot of cell types. Here, with the PhenoImager technology, we do a multispectral unmixing, where all the right colors come back to their right panel after the image has been taken, the informed image analysis software, and also we isolate autofluorescence. Here is an example where you can see there is a tissue autofluorescence masking a lot of the signal, but once you do the autofluorescence isolation, you can see the weakly expressing markers better. And also, you do the unmixing, you can see there is overlap. You remove the overlap and the bleed through, you can better see the marker expression. And it's not only a beautiful image, it gives you a better analysis, increases the signal to noise ratio. This is just an example of a whole slide multispectral imaging done using PhenoImager HT. You can see FOXP3, PAN-CK, PDL1, CD68, PD1, CD8. The PhenoImager HT, it can do 80 slides at a time. So you load them up in the hotel. It can do bright field and fluorescence imaging. And you can select whether it will do the bright field imaging on a slide carrier or a fluorescent or alternate. And it can do 6-plex within 10 minutes. This is an example of a bright field image. And the great thing is that even with bright field imaging, if you want to do spectral unmixing, you can do that if you have library slides for them. You can do multispectral IHC, you can see right here, where you can look at all these different stains and do a better counting or thresholding of these samples. These images are also compatible to be worked on other softwares, but you have to do the unmixing. If you look at this autofluorescence here, after unmixing, the autofluorescence is taken off, and you can do the whole slide scanning. And HT has a built-in TMA workflow, so it recognizes the cores as circular, and it can do the TMA scanning. And you can still do RNA-scope imaging on HT because it will remove the haze. And then there is a great fun feature that our informed machine learning software has. We call it pathology view. It creates a pseudo view of single markers from their fluorescent images. So a lot of people like that because they are used to seeing this kind of IHC views. So first you put in the raw image into the informed software. It will do the unmixing. Then maybe you will draw that this is tumor, this is stroma, and it will learn from your hand-drawn and create. Then you use the nuclear, cytoplasmic, and membrane signals to create the cell segmentation map. Based on the cell segmentation map, you can do an intelligent phenotyping for single markers or complex phenotypes. You can do age scoring with a 4 binning, 10 binning, or 50 binning. And finally, then you can look at all this analysis in this kind of spatial graphical format where you have upset plots, you have nearest neighbor analysis, you can look at the heat maps of the interaction. And when we get this technology out into the hands of researchers like you, we love to see them getting featured in the journal covers because they are pretty pictures and they look good. So today I've reached the end of my talk. The only thought I want to leave you all with is what will you do with your spatial phenotyping? That's the important question. Whenever somebody comes to us, we ask them this, what do you want to do? And how this technology can help you. Thank you very much for being present here and also virtually. I'll be happy to take any questions. Yeah. Hi. Sure. Polaris, yes. Yeah. It's just a rebranding. Exactly, same. There is no change in the instrument. It's the same thing. It's just our marketing team thought that phenotyping is a cool buzzword. Yeah, so sorry, if the so transition is that the codex with RNAscope is our collaboration with ACD. Then the next one is our proprietary chemistry that is coming out later this year, where you can use probes from us to do hundreds of RNA and protein on the same tissue. Technical transition would be the same. It will be using same barcodes and barcode technology and cyclical imaging as it does for the protein. Only, I think the limiting factor for us is we are like a proteomic company, like we come from the protein approach. So that's why we are not starting with a huge panel initially. We are starting with the one that you can do in a measurable amount of time so that you don't have to wait for weeks to know like thousands of things. So we have seen that a lot of our early adopters who have used this technology, they found that once they have done some orthogonal technologies, they can do the visualization with this many markers, say 50 proteins, 50 RNA, and they get a revealing result. Yeah. Thank you.
So practically, so say someone from our community wanted to get started, what steps would you recommend?
So for the pre-cooked panels, you just buy the panel and it comes with recommendation for different tissue types and you start. We have like discovery panels, which would be a 15-plex panel to deploy on your tissue. And you can start with that. Yeah, if you have access to the PhenoCycler system.
Question from one to take images live, but around 20 antibody panel.
20 antibody panel would be 5 hours based on the tissue size. Yeah. Because it includes both the fluidics and the imaging time. So we have like a proof of principle program called STEP, which is called Spatial Tissue Exploration Program, where we have some fixed panels. You can send in the tissue for a fee. We can run that for you. Or if you, yeah, we can run that for you and we do the analysis and give you back the data. So there are CROs and other cores that you can always avail, and I think Lily would be a better person to offline give you that information. Yes. Yeah. So you do the PhenoCycler, and then you do HU.
Thanks so much, Dr. Booker, for your time.
Thank you.
Technology.
Thank you. Do you want to come here and say that so that people in Zoom can? Yeah.
I just wanted to mention that because Dr. Haram is Jason worked with that, so it's gotta be really nice. Should I just go ahead? Yeah, so yeah, it hasn't been finalized with details, but I was saying we would do a grant, thank you, do a grant award here on campus through the core facility with Dr. Harms's group at the Rogel Cancer Center. And for some reason, the title of their core facility escapes me, so I apologize. But anyway, so AKOYA will donate some kits to have some multiplex staining done, and services can be done through the Rogel Cancer Center. Good.
We'll forward that. Cool. Other question? Great, thanks so much. Really appreciate it. Thank you for joining us. Next up.